In the previous videos, we discussed about the general introduction to genetics as well as contributions of Mendel's work to the genetics and the Mendelian principles and the laws. So, preferably the Mendelian laws or the principles are like first one, that is the principle of unit factor. Secondly, that is the law of dominance. Thirdly, that is the law of segregation or purity of gametes, and lastly, that is the law of independent assortment. And also we understood about how exactly the monohybrid cross and the dihybrid cross experiments could be carried out, and they were carried out by Mendel during his times. Based on this, let us try to understand one more new concept, that is interaction of genes. Here we need to discuss many aspects of it, and in brief I want to repeat a few most essential factors that are helpful for understanding it. The firstly, that is gene and alleles. Gene and allele. What is the difference between these two? First and the foremost, I want to tell you that a gene is a kind of chemical factor that helps in the expression of a trait, whereas allele is, or alleles are the variants of a gene. Alleles are the variations of a gene. Alleles are alternative forms of a gene. These alternative forms may be two or more than two. And however, alleles are to express a character. So now you must be knowing it well. Then what is the difference between trait and character? For example, that is the color of a seed. If this is a trait, and then its characters could be either yellow or green. Look at it. Color of the seed, when it is a trait, then its alternative expressions by the alleles are called as characters. Characters could be, color of the seed could be either yellow or green. I hope you followed this. This is very much essential. Then where do we find these genes or the alleles? So as to understand, we need to consider homologous chromosomes. Always chromosomes are present in pair in a diploid state, and here by referring this we can understand exactly how the gene and or the alleles are located. So let me draw quickly pair of chromosomes in a diploid set or diploid condition. In what condition we call these chromosomes as homologous? Homologous chromosomes, as I said, they are present in pair because one must be belonging to male parent as a paternal chromosome, another one chromosome belonging to female parent, so it is maternal chromosome. And to say that these paternal and maternal chromosomes are homologous to each other, they should have the similar location of a centromere, having equal sized arms. And here what you need to know is that each chromosome is, since it is formed of DNA, it is carrying the genes. The number of genes carried by paternal chromosome, if it is equal to the number of chromosomes carried by maternal chromosome, then these two are considered to be homologous pair. They are said to be more or less identical, but not 100%. Please try to underline this statement. Then now I want to show you, for example, how and where the genes are located. Here I show you, this is the second position. Let us say this is the second position in paternal and maternal chromosome for a gene, that gene to express color of the seed. If you consider, this could be yellow. This is an allele of a gene for color of the seed. This is yellow. Then that is present on the paternal chromosome. Then on the maternal chromosome, you need to have a corresponding allele to color of the seed. It may be either yellow or it may be green. It depends. I write capital Y, which is for yellow. It may be present on the maternal chromosome, or else allele for green color could be present. So now these are said to be Y gene, Y allele, allele present on paternal chromosome for yellow color is having a locus similar to that allele for green color is present. That means both these two alleles of a gene for color of the seed are belonging to the same locus. What do you mean by locus? This is very much important. Locus is the point exactly where gene is present. Locus is the position where exactly the gene is present on the chromosome only, because you need to see the genes only the chromosome, since they are made up of DNA material. Okay, let us say one more example, one more allele. Here is one allele for shape of the seed. We referred it in the previous experiment, hybrid cross experiment, where that is the shape of the seed. Either it could be allele for round shape of the seed, or it could be small r, which is allele present on maternal chromosome corresponding to this, where this can express a wrinkle shape of seed, or it may also contain capital R. It depends. So if the capital R present on the paternal chromosome, corresponding to that on the maternal chromosome, we find either capital R or small r. If we find capital R, that could represent a round seed. If it is a small r, look at it, it is a recessive, and that could be for wrinkle. However, these two alleles for shape of the seed are belonging to the same locus. This is my point to tell you. Please focus on this. One more trait let us consider, that is height of the stem in pea plant. Here is an allele for expressing height as tall. Let me represent it by capital letter T, and corresponding to that on maternal chromosome is an allele to be present for expressing height. It may be small t for expressing dwarf. In that place we may also find, instead of small t, capital T for tallness. Okay, capital T is for tallness, small t is for dwarfness. Look at it. This is the way how exactly for each gene to express a particular or specific character, we find two alleles belonging to the same locus. If we consider the position of alleles for expressing color of the seed is second position on paternal chromosome, on maternal chromosome must be also for second position, not for first or third or 10th or 11th, not like that. Similarly, if we consider another set of alleles for expressing the shape of the seed, look at it, capital R present on paternal chromosome with the position that is a 10th. If it is at position 10, this must be also at the position 10 on maternal chromosome. That means position of alleles of a gene must be located on the same locus in homologous chromosomes. This is said to be like these are intra-alleles. These are intra-alleles. alleles of the gene expressing a phenotypical character are belonging to the same locus. Like this we could say. Here one more example. The position of the alleles for a gene expressing height of the stem in the plant, position may be 19th, okay, on paternal chromosome. Corresponding to that, allele for the same trait, okay, for expressing height of the stem must be also present on the same position in its maternal chromosome. This is a way how exactly we could say that these alleles are belonging to the same locus. This is most important to understand. Okay, so I hope now you understood or you followed the meaning of homologous chromosomes. You may ask me a question that how many genes could be present on each in homologous pair of chromosomes. It depends on quantity of the DNA present in forming that set of chromosomes. Okay, and the next point is condition of the genes. We need to use two terminologies as homozygous condition, homozygous, and heterozygous. Let me make it very clear. It is sort of repetition now. Heterozygous. Okay, let me tell you homozygous. If for a particular trait, for a particular phenotype to express, if the alleles are, if they are found like capital Y for phenotype like color of the seed, then for shape of the seed, if it is like capital R, that is for round. Then height of the stem, if it is capital T, okay. Then these two alleles of a gene to express color of the seed are in homozygous state. Here capital R are the alleles are in homozygous state to express a phenotype that is round shaped seed. Then capital T, they are homozygous in the state or in nature because they express tallness in case of height of the stem, okay, height of the stem. Similarly, it could be also like this, like small y, or small r, or one more, that is small t. Then also these are homozygous in state, but they are recessive alleles. If we consider capital Y, capital T are homozygous dominant alleles, then small y, small r, small t are also in homozygous state to express their recessive character, recessive phenotype. This way, these are dominant homozygous state or dominant recessive homozygous state. Then for what we call it as heterozygous condition. If for example color of the seed, the genotype, if it is present like this, one allele is capital Y, another allele is small y, look at it here, these are in heterozygous state because this allele is for yellow color, this allele is for green color. Still then character expressed here is yellow colored seed, but they are heterozygous in state. Here also express yellow color, but here also express yellow color, but this is homozygous and this is heterozygous. Similarly, it could be like this for shape of the seed, like capital R, small r for expressing what? That is a round shape. Here homozygous round shape, here heterozygous round shape. Then this could be like capital T, small t. This is heterozygous for tallness. Here also heterozygous for tallness. But we can never find recessive alleles expressing their respective character or a phenotype in heterozygous state. Always in heterozygous state, allelic pair when we consider, one allele will express which is found to be always dominant by suppressing the other. So this is a way how exactly we have understood homologous chromosomes means what, then homozygous and heterozygous condition of the allelic pair or the genes on a chromosome belonging to the specific locus. We have understood. After knowing this, now coming to the main point, that is the main concept that we need to know or we need to study today is interaction of genes. Let me rub this and Interaction of genes. Genes may be belonging to the same loci, same locus or a different locus. They are supposed to interact with each other. That's what we call it as interaction of genes. Please remember, to understand this, always we need to consider homologous chromosomes only, homologous pair only, okay? And interaction of genes we could observe in two different conditions, okay? First type, that is called as intragenic or intra-allelic interaction. Another type, that is intergenic or interallelic. Please try to differentiate. Interallelic, that is intra, and this is interallelic interaction. There are two types, capital A, this is capital B. Second type, first type, second type. Intragenic, intraallelic, then intergenic or interallelic, second type. Then how do we understand it? For that reason, once again, we need to consider homologous pair of chromosomes. Homologous pair ಅಂದ್ರೆ ಕ್ಲಿಯರ್ ಆಗಿ ಗೊತ್ತಿರಲಿ. ಒಂದು ಜೋಡಿ ಅಂದ್ರೆ ಒಂದು ಪೇರೆಂಟಲ್ ಮೇಲ್ ಪೇರೆಂಟಲ್ ಕ್ರೋಮೋಸೋಮ್ ಇರ್ತದೆ, ಇನ್ನೊಂದು ಫೀಮೇಲ್ ಪೇರೆಂಟಲ್ ಕ್ರೋಮೋಸೋಮ್ ಇರ್ತದೆ. ಓಕೆ, ದಿಸ್ paternal, this maternal. Okay, now so as to understand, intragenic or intraallelic means what? Just we will take the same example what we considered previously, that is the color of the seed. Here if this is capital Y, dominant allele for expressing yellow color, then if recessive allele for expressing wrinkle color present on the same locus of the maternal chromosome, look at it. These two alleles, dominant and recessive allele for expressing color of the seed, are belonging to the same locus because their position is same on their respective paternal and maternal chromosome. Please understand it. Secondly, now what we need to know, that this capital Y, if it interacts with this, if it interacts, capital Y interacts with the small y, means a dominant allele when it interacts with the small allele, then that would form a genotype as capital Y, small y, and this obviously could express a character that is yellow only. Look at it as per law of dominance. If one more example I give you now, that is height of the cell in pea plant. It may be capital T on paternal chromosome, small t on maternal chromosome. Look at it. This capital T dominant allele, if it interacts with this small t allele belonging to the same loci, then this could express like capital T small t for tallness only. But the thing is that how yellow color seed and tallness of the pea plant are expressed. Look at it. This is because intra-allelic interaction. Intra-allelic interaction means capital Y interacted with small y for expressing yellow color. This is called as intra-allelic or intragenic interaction. If this capital T interacts with small t, then it is called intra-allelic interaction or intragenic interaction. Okay, this is how exactly, and we can take many examples like where do we find intragenic or intraallelic interaction? We can find it in case of incomplete. This is another type of expression of phenotype, incomplete dominance, where it is a case of intraallelic interaction. Then one more is codominance. Codominance. Then the third one, multiple allelism. In all these three cases, we find intragenic or intraallelic interaction. Intraallelic interaction is the one where alleles belonging to the same loci interact among themselves to express a specific character. Okay, so that's all about intra-allelic. Now, something different. What is it? Instead of, if we consider this is a dominant allele for color of the seed, this is a dominant allele for height of the stem in pea plant. If this dominant allele interacts with this, or this dominant allele interacts with this, which are belonging to the different locus. ಇದರದ್ದು ಪೊಸಿಷನ್ 2 ಇದೆ ಅಂತ ಅನ್ಕೊಂಡಿದ್ದೀವಿ ಅಂದ್ರೆ ಇದರದ್ದು ಪೊಸಿಷನ್ 19 ಐತೆ ಅಂತ ಅನ್ಕೊಂಡಿದ್ದೀವಿ ಅಂತಂದ್ರೆ look at it. See, capital Y belonging to the locus 2 and capital T belonging to the locus 19. For example, I am telling you, when these two interact together, which are not, the loci are different, position of these two dominant alleles are different, okay, is different. So this is a case of intergenic or interallelic interaction. Instead of acting its corresponding allele belong to the same locus, here dominant alleles, particularly dominant alleles belonging to the different locus, if they interact, this is called as intergenic or intra-allelic interaction. I hope you people followed it correctly. And for this type of concept, could we give examples? Yes, there are many. First example that we need to consider is supplementary genes or supplementary alleles. Example. So this could be the best example for understanding how alleles belong to the different loci interact together to express the same phenotype. Next one could be complementary genes or complementary factors. Third could be dominant epistatic genes or factors. There could be one more, that is a dominant epistatic, it is not dominant, it is recessive. It is recessive epistatic genes or the factors. Fifth one, that is lethal factors. We will discuss and understand each one of these 5 one by one very clearly, along with the examples, along with the experiments that are carried out in the different species of animals. Okay, so this is the set of examples for understanding intergenic or interallelic interaction, and this is a set of examples for understanding intragenic or intraallelic interaction. So okay, and we will discuss all this in the next topic, but in this particular topic we are expected to discuss in detail along with the examples of these, like supplementary, complementary genes, then dominant and recessive epistatic genes, then lethal genes, etc. Today we shall consider the supplementary genes so as to understand interallelic or intergenic interaction. For this reason, we need to take, first let me write supplementary genes or factors. We have to take this as an example. Okay, for this let us understand the concept of these supplementary genes. Here is once again homologous paired chromosomes. Okay, supplementary genes means there must be 2 dominant genes to express a single phenotype. that is here. Let me say this is capital A for some to express particular phenotype. This could be small a, and here is on another locus of the homologous chromosomes. Here is a capital B and a small b to express another one. What is that? Phenotype. If these two are considered to be supplementary, they have to interact with each other. Capital B gene, dominant gene, must interact with the capital A. But before that, let me tell you nature of this capital A allele. Capital A allele is able to express a character independently means it need not depend on expression of capital B dominant gene. Then capital B, nature of capital B, another one dominant gene belong to different locus, is unable to express a character independently, but it has ability. This capital B as a dominant, second dominant allele has ability to modify or modulate the expression of capital A. That means this will give support to capital A to express a different kind of character than the normal character. How is this possible? This is possible because we can witness this type of interaction as a supplementary genes interaction in case of what is it? Comb shape or comb pattern in fowls. You know, fowl, it could be cock or hen. Fowl anta ನಾವು ಕರೀತೀವಿ. ಸೊ ಇಲ್ಲಿ ಸಪ್ಲಿಮೆಂಟರಿ ಕನ್ನಡದಲ್ಲಿ ಹೇಳಬೇಕು ಅಂತ ಸಪ್ಲಿಮೆಂಟರಿ ಜೀನ್ಸ್ ಅಂತ ಯಾಕೆ ಕರಿಬೇಕು ನಾವು? ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ನೋಡ್ರಿ, ಕ್ಯಾಪಿಟಲ್ ಎ ಮತ್ತು ಕ್ಯಾಪಿಟಲ್ ಬಿ ಎರಡು ಜನರಲ್ ಆಗಿ ಡಾಮಿನೆಂಟ್ ಅಲೀಲ್ಸ್ ಅದಾವ ಅಂತ ಅನ್ಕೊಳ್ರಿ. ಕ್ಯಾಪಿಟಲ್ ಎ ಒಂದು ಯಾವುದೋ ಪರ್ಟಿಕ್ಯುಲರ್ ಫಿನೋಟಿಪಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡಲಿಕ್ಕಿದೆ. ಕ್ಯಾಪಿಟಲ್ ಬಿ ಇದು ಕೂಡ ಡಾಮಿನೆಂಟ್ ಅಲೀಲ್, ಬಟ್ ಅದು ಬೇರೆ ಲೋಕಸ್ ಮೇಲೆ ಇದೆ, ಇನ್ನು ಹೋಮೋಲಾಗಸ್ ಕ್ರೋಮೋಸೋಮ್. ಹಿಂಗಿರ್ಬೇಕಾದ್ರೆ ಈಗ ಕ್ಯಾಪಿಟಲ್ ಬಿ ನಾವು ಕ್ಯಾಪಿಟಲ್ ಎ ಸಪ್ಲಿಮೆಂಟರಿ ಜೀನ್ಸ್ ಅಂತ ಯಾಕೆ ಕರಿಬೇಕು? ಜಸ್ಟ್ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ತಗೊಳ್ಳಿ. ಎಕ್ಸಾಮ್ನಲ್ಲಿ ನೀವು ಮೇನ್ ಆನ್ಸರ್ ಪೇಪರ್ ತಗೊಳ್ತಿದ್ರಿ. ಮೇನ್ ಆನ್ಸರ್ ಪೇಪರ್ನಲ್ಲಿ ಸುಮಾರು ನಿಮ್ಗೆ 28 ಪೇಜ್ ಇರ್ತಿದೆ ಅಂತ ನೀವು ಕನ್ಸಿಡರ್ ಮಾಡಿಕೊಂಡ್ರೆ ಅದಕ್ಕೆಲ್ಲ ನೀವು ಬರೀತಾ ಹೋಗ್ತೀರಿ. ಓಕೆ, ಆಮೇಲೆ ಒಂದು ಇನ್ನೂ ಅಷ್ಟು ನಿಮಗೆ ಕ್ವಶನ್ಸ್ಗೆ ಉತ್ತರ ಆನ್ಸರ್ ಬರೆಯೋದಿರ್ತದೆ. ಅವಾಗ ನಿಮ್ಗೆ ಎಕ್ಸ್ಟ್ರಾ ಪೇಜ್ ಬೇಕು ಅಂದ್ರೆ ಅವಾಗ ನೀವು ಒಂದು ರಿಕ್ವೆಸ್ಟ್ ಮಾಡ್ಕೋತೀರಿ ಇನ್ವಿಜಿಲೇಟರ್ನ, ಅದನ್ನು ಸಪ್ಲಿಮೆಂಟ್ ಬೇಕು ಅಂತ ಹೇಳಿ. ಸಪ್ಲಿಮೆಂಟ್ ನೀವು ತಗೋತೀರಿ. ಲುಕ್ ಅಟ್ ಇಟ್, ಸಪ್ಲಿಮೆಂಟ್ ಮೀನ್ಸ್ ಆ ಸಪ್ಲಿಮೆಂಟ್ ತೊಗೊಂಡು ನೀವು ಇನ್ನುಳಿದಂತಹ ಏನು ಮಾಡ್ತಿದ್ರಿ, ಕ್ವಶನ್ಸ್ಗೆ ಆನ್ಸರ್ಸ್ ಬರೀತಿದ್ರಿ. ಆಮೇಲೆ ಸಪ್ಲಿಮೆಂಟ್ ಏನು ಮಾಡ್ತೀರಿ, ತೆಗೆದೋಗಿಯಲ್ಲ, ಅದನ್ನು ತಗೊಂಡು ಮೇನ್ ಆನ್ಸರ್ ಪೇಪರ್ಗೆ ಟ್ಯಾಗ್ದಿಂದ ಗಂಟು ಕಟ್ತೀರಿ. ಓಕೆ, ಸೊ ನಾವು ಏನಾಗುತ್ತೆ, ವ್ಯಾಲ್ಯೂಯೇಟರ್ ಎಕ್ಸಾಮ್ ವ್ಯಾಲ್ಯೂಯೇಷನ್ ಮಾಡೋ, ಪೇಪರ್ ವ್ಯಾಲ್ಯೂಯೇಷನ್ ಮಾಡುವಂಥ ಪ್ರೊಫೆಸರ್ ಏನು ಮಾಡ್ತಾರೆ, ಫಸ್ಟ್ ನಿಮ್ಮ ಮೇನ್ ಆನ್ಸರ್ ಪೇಪರ್ದೆಲ್ಲ ಚೆಕ್ ಮಾಡ್ತಾ ಹೋಗ್ತಾರೆ. ಸಪ್ಲಿಮೆಂಟ್ ಬರ್ತದೆ, ಅಲ್ಲಿಯೂ ಕೂಡ ಚೆಕ್ ಮಾಡ್ತಾರೆ. ಅದಕ್ಕೆ ಬಂದಂಥ ಮಾರ್ಕ್ಸನ್ನು ಇದಕ್ಕೆ ಆ್ಯಡ್ ಮಾಡ್ತಾರೆ. ಲುಕ್ ಅಟ್ ಇಟ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಮೇನ್ ಆನ್ಸರ್ ಪೇಪರಿಗೆ ಸುಮಾರು ನಿಮ್ಗೆ 65 ಮಾರ್ಕ್ಸ್ ಬಂದಿತ್ತು ಅಂತಂದರೆ ಸಪ್ಲಿಮೆಂಟಲ್ಲಿ ಬರೆದಂಥದ್ದಕ್ಕೂ ವ್ಯಾಲ್ಯೂಯೇಷನ್ ಮಾಡಿದಾಗ ಸುಮಾರು ಅದಕ್ಕೊಂದು 15 ಮಾರ್ಕ್ಸ್ ಬಂದಿತ್ತು ಅಂತಂದರೆ ಅದು ಇದು ಕೂಡಿಸಿದ್ರೆ ಟೋಟಲ್ ನಂಬರ್ ಆಫ್ ಮಾರ್ಕ್ಸ್ ಎಷ್ಟಾಗ್ತದೆ? 65 ಪ್ಲಸ್ 15 ಟೋಟಲಿ 80 ತಗೊಳ್ಳೋರಾಗ್ತಿದೆ. This is how the main answer paper number, total number is modified by considering the score of your supplement paper. ಹಂಗನ ಇಲ್ಲಿ ಮೇನ್ ಏನು ಅಂತ ಇಂಪಾರ್ಟೆಂಟ್, ನಿಮ್ದು ಮೇನ್ ಆನ್ಸರ್ ಪೇಪರ್ ಇದ್ದಾಗ ಕ್ಯಾಪಿಟಲ್ ಎ ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಅಂತಂದ್ರೆ, ಆದರೆ ಕ್ಯಾಪಿಟಲ್ ಬಿ ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಏನು ಅಂತಂದ್ರೆ ಇದು ನೀವು ಬೇಕಾದ್ರೆ ಸಪ್ಲಿಮೆಂಟ್ ತಗೊಳ್ಳಿ, ಬೇಕಾದ್ರೆ ಬಿಡ್ರಿ, ನಿಮಗೆ ಮಾರ್ಕ್ಸ್ ಬಂದೇ ಬರ್ತದೆ. ಆದರೆ ಇನ್ನೊಂದು ಏನು, ಇಲ್ಲಿ ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಬಿ ತೊಗೊಂಡಿವಿ ಅಂತಂದ್ರೆ ಇಂಡಿಪೆಂಡೆಂಟ್ ಆಗಿ ಅದು ಕೆಲಸ ಆಗಲ್ಲ. ಬರೀ ನೀವು ಆನ್ಸರ್ ಪೇಪರ್ ತೆಗೆದು ಕೊಟ್ಟು ಬರೀ ಸಪ್ಲಿಮೆಂಟ್ ಒಂದು ನೀವು ಸಬ್ಮಿಟ್ ಮಾಡಿದರೆ ಮಾರ್ಕ್ಸ್ ಬರಲ್ಲ. ಇಟ್ ಈಸ್ ನಾಟ್ ವ್ಯಾಲಿಡ್, ಯುವರ್ ರಿಸಲ್ಟ್ ವಿಲ್ ಕಮ್ ಫೇಲ್. ಹಂಗೆ ಕ್ಯಾಪಿಟಲ್ ಬಿ ಇಂಡಿಪೆಂಡೆಂಟ್ ಆಗಿ ಕೆಲಸ ಮಾಡಲ್ಲ. ಇಟ್ ವಿಲ್ ಹ್ಯಾವ್ ಟು ವರ್ಕ್ ವಿತ್ ಕ್ಯಾಪಿಟಲ್ ಎ. ಕ್ಯಾಪಿಟಲ್ ಎ ಬಿ ಹರ್ಕತ್ತಿಲ್ಲ. ಆದರೆ ಕ್ಯಾಪಿಟಲ್ ಬಿ ಇತ್ತು ಅಂತಂದ್ರೆ, ಅದು ಇಂಟ್ರಾಕ್ಟ್ ಮಾಡಕ್ಕೆ ಬಂತು ಅಂತಂದ್ರೆ, ಕ್ಯಾಪಿಟಲ್ ಎ ತನ್ನ ಒಂದು ಏನು ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡ್ತಿರ್ತದೆ ಕ್ಯಾರೆಕ್ಟರ್, ಅದನ್ನು ಇದು modify ಮಾಡ್ತಿದೆ. That's why it is called as supplement. These two are called as supplementary genes. Okay, and let us give one practical evidence to this by considering ಏನಂತ comb shape or pattern, comb shape or pattern in fowl. ನೀವು ಬೇರೆ ಬೇರೆ ಫೌಲ್ ಬ್ರೀಡ್ಸ್ನ ಹೋದ ವರ್ಷ ಕಲ್ತಿದ್ದೀರಿ. ಅದ್ರಾಗಿನೂ ಕೋಂಬ್ ಅಂದ್ರೆ ಗೊತ್ತಾ, ಅದಕ್ಕೆ ನಾವು ಕನ್ನಡದೊಳಗೆ ತಲೆ ಮೇಲೆ ತುರಾಯಿ ಅಂತ ಕರಿತೀವಿ. ದಟ್ ಈಸ್ ಕೋಂಬ್. ಓಕೆ, ದಟ್ ಈಸ್ ಯೂಶಲಿ ರೆಡಿಶ್ ಕಲರ್ ಇರ್ತದೆ ಕಾಕ್ನಲ್ಲಿ, ಹೆಣ್ಣಲ್ಲೂ ಇರ್ತದೆ. ಕೆಳಗೆ ಒಂದು ಏನು ಬಂದಿರ್ತದೆ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ವ್ಯಾಟಲ್, ಬಟ್ ಡೋಂಟ್ ಗೆಟ್ ಕನ್ಫ್ಯೂಸ್ ವಿತ್ ದಿಸ್. ಓಕೆ, ಸೊ comb shape or comb pattern. Let me tell you, there is a gene to express a trait. That trait is comb shape or comb pattern in fowl. But this gene will be having alternative allelomorphs. Okay, and how many pattern of fowl, combs we find in fowl breeds? Let me give you one example. That is in case of Vandote breed. Otherwise I'll get it here. So time is limited. Vandote breed. It is a kind of fowl breed. Vandote breed. In this case, the type of comb pattern we find is said to be like a rose comb. Then one more example, let us take a Brahma breed. We find a pea comb. Then one more example, that is Leghorn. You must have studied white Leghorn breed. In this case we find not rose or pea comb type. It is different, called single comb. There is one more breed I write, but I will write question mark before it. That is walnut breed. In this case, type of comb pattern is now in question. We will answer it by solving this. Okay, to express this rose comb pattern in Vandote, definitely this is a kind of, look at it, a trait, and it is expressed by a gene called as capital R. Then the pea comb in Brahma breed is expressed by another gene, that is capital P. Single comb, here it is expressed by the recessive form of capital R and capital P. They are small r and small p. Whereas one more kind of comb pattern, it is expressed by both capital R and capital P together. Then please, one thing I request you to differentiate. Dihybrid cross experiment in case of Mendel's work. Look at it, where we considered two genes belonging to the different loci, but express two different phenotypical characters. For example, ali color of the seed adkondu gene, then shape of the seed adkondu gene. Note, shape of the seed ಮತ್ತು color of the seed ಎರಡು genes ಎರಡು characters ಬರ್ತಿದೆ. ಅಲ್ಲಿ ಕಲರ್ ಆಫ್ ದ ಸೀಡ್ ಬಂತು ಅಂತಂದ್ರೆ ಅದು ಯೆಲ್ಲೋ ಆದ್ರೂ ಆಗ್ಬೋದು ಅಥವಾ ಗ್ರೀನ್ ಆದ್ರೂ ಆಗ್ಬೋದು. ದೆನ್ ಶೇಪ್ ಆಫ್ ದ ಸೀಡ್ ನೋಡಿದ್ರೆ ಅದು ರೌಂಡ್ ಆದ್ರೂ ಆಗ್ಬೋದು ಅಥವಾ ರೆಗ್ಯುಲರ್ ಆದ್ರೂ ಆಗ್ಬೋದು. ಬಟ್ ಇಲ್ಲಿ ಟ್ರೈ ಮಾಡ್ರಿ, ಇಲ್ಲಿಯೂ ಕೂಡ ಎರಡು ಜೀನ್ಸ್ ಪ್ಯಾರಲಲ್ ಆಗಿ ವರ್ಕ್ ಮಾಡ್ತವೆ. ದಟ್ ಈಸ್ ಸಪ್ಲಿಮೆಂಟರಿ ಫ್ಯಾಕ್ಟರ್ಸ್, ನಾಟ್ ಫ್ಯಾಕ್ಟರ್. ಸಪ್ಲಿಮೆಂಟರಿ ಫ್ಯಾಕ್ಟರ್ ಆಗಲಿದೆ ಕ್ಯಾಪಿಟಲ್ ಎ ಕ್ಯಾಪಿಟಲ್ ಬಿ. ಹಂಗೆ ಇಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಅಂಡ್ ಕ್ಯಾಪಿಟಲ್ ಪಿ, these two are the two dominant genes present at the different position in the homologous paired chromosomes, but they tend to interact with each other. That is intergenic interaction. Position ಬೇರೆ ಬೇರೆ ಕಾರಣಕ್ಕೆ. ಓಕೆ, ಸೊ ಹೀಗಿರಬೇಕಾದ್ರೆ ನಾನು ಹೇಳಿದೆ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಫಾರ್ ರೋಸ್ comb is dominant, capital P for pea comb is dominant. How do I prove it? We can prove it by crossing rose combed fowl with the single combed leghorn, because it is recessive as I said before. Okay, let me write case 1. Okay, case 1. If we cross rose combed fowl with single combed leghorn, rose combed Vandote breed, if we cross it with single combed leghorn. But please remember, rose comb, this must be male. This must be male. Andre idu cock irbeku, idu hen irbeku. Andre adukku cross anta ನಾವು ಕರಿತೇವೆ. Cock and cock cannot interbreed. Then hen and hen cannot interbreed. Cock and hen, male and female should, they should, we can do the crossing. Okay? And rose comb, the particular trait, the phenotype, genotype ಅನ್ನು ಬರೆಯೋಣ. ರೋಸ್ comb ಅನ್ನೋದು ಫಿನೋಟೈಪ್ ಆದರೆ ಅದರ ಜಿನೋಟೈಪ್ ನಾವು ಹೇಗೆ ಬರೆಯಬಹುದು? ಕ್ಯಾಪಿಟಲ್ R, small p. Please remember, small p represents the recessive allele of a dominant P, capital P. Okay? Then here, single comb, we need to write alleles of capital R and capital P are in recessive state, so it is small r, small p. Okay? And from this gametes are produced, from this sperms are produced, these are sperm. What I am going to write? Capital R, small p. So we need to keep it in circle. And small here, small r, small p. This is egg or ovum produced by single combed fowl. Okay? Then only this sperm can unite with this ovum or egg. Okay? So when these two unite together, sperm of rose combed fowl unites with what? That is a single comb of this what is it? Which of leghorn. When these two sperm and egg are united, they together form a genotype, capital R, small r, then small p. Look at it. Capital R gene is for rose comb, whereas small r, no. Then small p, they are not for single comb. Okay? So that obviously this is considered as F1 generation, and the phenotype of this genotype is, that is the rose comb only. Means, ಇದರಿಂದ ಹುಟ್ಟಿದಂತಹ ಎಷ್ಟು total number of chicken that are reproduced by crossing rose comb and single combed fowl are found to be with the rose comb only, but not at all a single comb. By means of this we can prove that gene for rose comb is dominant over gene for single comb. Very clear. And once again, if you cross fowls of this type of what genotype, you will get, no doubt, 3/4 of it will be found with rose comb and 1/4 of it will be found with single comb. That is a 3:1 ratio according to your law of dominance, Mendelian principle, Mendelian law. That is there. Okay? This is simple crossing. It is a kind of crossing between the two supplementary genes possessed fowls. Then similarly, let us consider case 2, where to prove that gene for P comb is dominant. Case 2. Here I write P combed what? Brahma breed crossing with single combed leghorn. For P comb, you need to write a genotype, that is small r, capital P, where capital P is for P comb, and single comb, as you all know, recessive, small r, small p. And they produce their respective sperm and eggs. And here this every sperm produced by this P comb will be carrying genotype, that is small r, capital P, and a single combed fowl producing eggs carrying genotype, that is small r, small p. When these two are united, means fertilization is occurred, then its respective genotype will be small r, capital P. Look at it, capital P is for the expression of a phenotypical character as P comb only. That means gene for P comb is dominant over gene for single comb. This way, gene for rose comb and gene for P comb are dominant over single combed pattern in case of leghorn. Now we have proved. Once again, if you cross this by taking this F1 generation to get F2 generation, you will get 3/4 of the F2 generation will be found with P comb and 1/4 of the generation will be found with single comb. Again 3:1 classical ratio of monohybrid cross, where we proved once again that P combed fowl gene is dominant over single combed gene recessive. So that's how. And case 3. So as to understand now literally what is it? Case 3. I need more space for it, so I write it here. Case 3. ಇಲ್ಲಿವರೆಗೆ ನಾವು ರೋಸ್ ಕೋಂಬ್ನ ಸಿಂಗಲ್ ಕೋಂಬ್ನ ಫಾಲ್ ಜೋಡಿ ಕ್ರಾಸ್ ಮಾಡಿದ್ವಿ, ಪಿ ಕೋಂಬ್ನ ಫಾಲ್ನ ಸಿಂಗಲ್ ಕೋಂಬ್ನ ಫಾಲ್ ಜೋಡಿ ಕ್ರಾಸ್ ಮಾಡಿದ್ವಿ. ಇಲ್ಲಿ ಆರ್ ಅಂಡ್ ಪಿ ಡಾಮಿನೆಂಟ್ ಅಂತ ತಿಳ್ಕೊಂಡಿದ್ವಿ. ಬಟ್ ಈಗ ಇಫ್ ವಿ ಕ್ರಾಸ್ ರೋಸ್ ಕೋಂಬ್ಡ್ fowl with P combed fowl, once again it must be female and it must be male. This is a male symbol and this is female symbol. Please you follow it. Sorry, ಇದನ್ನ ಕ್ರಾಸ್ ಮಾಡಿದ್ರೆ ಏನಾಗ್ತದೆ? ಥರ್ಡ್ ಕೇಸ್ ಇದೀಗ. If when we cross this, let me write the genotype for rose comb as capital R, small p as you know, and for P comb that is a small r, capital P. So it is a genotype for rose comb and P comb respectively. And rose combed fowl, when it produces sperms, it carries a genotype. I mean alleles are, one is found dominant, another one is found recessive. ಇದರಿಂದ ಸಾವಿರ ಸ್ಪರ್ಮ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರೆ in each sperm it carries this kind of genes for respective traits. Sorry, trait, not traits. ಒಂದು trait ಇಲ್ಲಿ ಸ್ಮಾಲ್ r, ಕ್ಯಾಪಿಟಲ್ P. ಇದರಿಂದ ಎಷ್ಟೇ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಲಿ, 10 ಪ್ರೊಡ್ಯೂಸ್ ಆಗಲಿ, 20 ಪ್ರೊಡ್ಯೂಸ್ ಆಗಲಿ, each ovum should carry this type of genotype for expressing a trait called comb pattern. Okay, and sperms of this genotype, this type, and egg of this genotype, when they are fertilized, egg is fertilized, then it forms a genotype as capital R, capital P, small p. Please look at it. Capital R is the dominant, capital P is the dominant, but these two are, we are saying that these two are supplementary to each other. Neither we get phenotype for this genotype is rose or P comb. It is something different. We call it as walnut comb. In this F1 generation, what we get the phenotypical character expression is by genotype like capital R, capital P, small p, is something new, something different from rose comb and P comb. ಇಲ್ಲಿ ರೋಸ್ ಕೊಂಬನು ಅಪಿಯರ್ ಆಗಲಿಲ್ಲ, ಪಿ ಕೊಂಬನು ಅಪಿಯರ್ ಆಗಲಿಲ್ಲ. ಯಾಕಂದ್ರೆ ಕ್ಯಾಪಿಟಲ್ ಪಿ ಸಪ್ಲಿಮೆಂಟರಿ ಜೀನ್ ಟು ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಅನ್ನೋದಾದ್ರೆ ಕ್ಯಾಪಿಟಲ್ ಪಿ ಇದ್ದದ್ದು ಕ್ಯಾಪಿಟಲ್ ಆರ್ ದ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಅನ್ನ ಮಾಡಿಫೈ ಮಾಡ್ತು. ಅದಕ್ಕಾಗಿ ಇಲ್ಲಿ ನಾವು ವ್ಯಾಲೆಂಟ್ ಕೋಂಬ್ಡ್ ಫೌಲ್ ಜನರೇಷನ್ ಅನ್ನ ಕಂಡುಕೊಂಡಿದ್ದೀವಿ. All F1 individuals are with something new comb pattern character, that is a valent comb. You try to understand. This is a clear indication of the modification of one dominant gene by another dominant gene belonging to the different loci. That's why they are said to be supplementary to each other. Andre capital R is independent of capital P, no doubt, but capital P is not independent, but it can modify the expression of capital R. Then so as to get F2 generation, now ಏನು ಮಾಡಬಹುದು? We have to cross once again by taking F2 generation. What is it? Here male genotype carrying ಇದು walnut with walnut cross ಮಾಡೋಣ. ಒಂದು ವ್ಯಾಲ್ನಟ್ ಕೋಂಬ್ಡ್ ಫೌಲ್ ಮೇಲ್ ಇರಬೇಕು, ಇನ್ನೊಂದು ವ್ಯಾಲ್ನಟ್ ಕೋಂಬ್ಡ್ ಫೌಲ್ ಫಿಮೇಲ್ ಇರಬೇಕು. ಕಂಡೀಷನ್ Okay, and let me write genotype for it. The same genotype we need to take capital R, capital P, small p, and here also the genotype is same capital R, small p. What is it? Sorry, capital R, capital P, small p, right? And sperms produced by, if we consider this as a male and this as a female, this produces sperms and that produces eggs with the genotype. ಯಾವ possible combination of alleles ಏನೇನು ಬರ್ತಾವ ನೋಡಿ. Here 1/4 of the sperm produced by valent comb, male valent comb fowl is capital R, capital P. This is one possible combination. Another 1/4 set of sperms carrying genotype, that is capital R, small p. Gametes sperm anta circle alli idta idini. Then small r, capital P. This is another allelic combination. Another 1/4. Remaining 1/4 will be carrying genotype recessive small r, small p. Idu possibility. Hanga idrinda ನಾಲ್ಕು possible combinations ಆಗ್ತವೆ. Capital R, capital P, capital R, small p, small r, capital P, and last one small r, small p. Hinga. This is for probable condition considered. If this is permanent with this, what will be the combination? This with this, what will be the genotype? Here we need to take help of a checkerboard or a Punnett square board. Let us do it here. Punnett square board. Okay. We need to draw a table having columns and rows 60 squares. We need to form 1, 2, 3, 4, and here also rows need to be 4 as allelic combination are 4. We get totally 16 kinds of genotypes with their respective phenotype. Okay, and ಇಲ್ಲಿ ನಾನು ಬರೀತೇನೆ sperms ಅಂತ ಈ ಕಡೆ allelic combination capital R, capital P ಹೀಗೆ ಮಾಡ್ತೀನಿ, capital R, small p ಹೀಗೆ ಮಾಡ್ತೀನಿ, small r, capital P, and last one small r, small p. Let me represent with the different colors. ಒಂದನ್ನ ರೆಡ್ ಅನ್ನ, ಇನ್ನೊಂದನ್ನ this is with white and this is with exit. Capital R, small p, and one more, that is a small r, capital P, and ಇನ್ನೊಂದು ಕಲರ್ ಯೂಸ್ ಮಾಡ್ತೀನಿ small r, small p. ನಾಲ್ಕು ಕಲರ್ ಬೇರೆ ಬೇರೆ ಆಗುತ್ತೆ. ಅದೇ ರೀತಿ ಇದು ಕೂಡ ಈ ಕಡೆದು ಓವಮ್ ಅಥವಾ ಎಗ್. ಓಕೆ, ಇದ್ರದ್ದು ಜಿನೋಟೈಪ್ ಕೂಡ ಹೀಗೆ ಇರ್ತದೆ, ಕ್ಯಾಪಿಟಲ್ ಆರ್, ಕ್ಯಾಪಿಟಲ್ ಪಿ ಒಂದು, ಇನ್ನೊಂದು ಕ್ಯಾಪಿಟಲ್ ಆರ್, ಸ್ಮಾಲ್ ಪಿ, ಮತ್ತೊಂದು ಸ್ಮಾಲ್ ಆರ್, ಕ್ಯಾಪಿಟಲ್ ಪಿ, ದೆನ್ ಇನ್ನೊಂದು ಸ್ಮಾಲ್ ಆರ್, small p, right? So ಇದು ಕೂಡಿದರೆ when this is crossed with this capital R, okay, capital R, then capital P. If this here unites with this sperm genotype, ಏನಾಗ್ತಿದೆ capital R, capital P, small p, and if this sperm here unites with this sperm, this is capital R, small r, capital P, and this here unites with this kind, that is capital R, small r, capital P, small p. Okay, then second row, if this here unites with this capital R, capital P, small p, and here this is capital R, small p, and here this is capital R, small r, capital P, small p, and this one capital R, small p. Third row, capital R, capital P. Then this is with capital R, capital P, small p. And this is small r, capital P. And this is with small r, capital P, small p. And the last row, this is capital R, capital P, small p, capital P, small p. Then this is with capital R, small p. And this is with small r, capital P, small p. And last one, small r, small p. These are the totally 16 genotypes we get by representing the cross between walnut-combed male fowl with walnut-combed female fowl, cock and hen, with this combination. ಇದ್ರದ ಫಿನೋಟೈಪ್ ಅನ್ನು ನಾವು ಈಗ ರಿಪ್ರೆಸೆಂಟ್ ಮಾಡೋಣ. ಎಲ್ಲೆಲ್ಲಿ ನಮಗೆ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಕ್ಯಾಪಿಟಲ್ ಪಿ ಬರ್ತಾವೋ ಅದು ಯಾವಾಗಲೂ ವ್ಯಾಲ್ನಟ್ ಆಗ್ತದೆ. ಸೊ ಇದನ್ನು ನಾವು ಹೇಳ್ತೀನಿ. ವಾಟ್ ಈಸ್ ಇಟ್? Capital R, capital P, walnut, because all this row entirely containing genotype with capital R and capital P, all these four are inherited, that is walnut. Understand? ಸ್ವಲ್ಪ ಫಾಲೋ ಮಾಡಿ. ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಕ್ಯಾಪಿಟಲ್ ಪಿ ಜಿನೋಟೈಪ್ ನೇ ಬಂತು ಅಂದ್ರೆ ವ್ಯಾಲೆಂಟ್ ಆಗಲಿಕ್ಕೆ ಬೇಕು. ಓಕೆ, ಬಿಕಾಸ್ ಬೋತ್ ಆರ್ ಸಪ್ಲಿಮೆಂಟರಿ ಟು ಈಚ್ ಅದರ್. ಅಂಡ್ ಇಲ್ಲಿ ನೋಡಿ, ದಿಸ್ ಈಸ್ ವ್ಯಾಲೆಂಟ್, then this is valent. In this row, how many? 2 valents. Then third row dal nodri, this is valent, yes, only 2. Here also 2 valents. Then e row dal nodri, this is valent and this is only valent, and here is 1 valent. Totally ಎಷ್ಟು ಆದ ನೋಡ್ರಿ, 4 + 2 6 + 2 + 1, ಟೋಟಲಿ 9 ವ್ಯಾಲೆಂಟ್ ಆದವು. ಈಗ ಇನ್ನೊಂದು ಕಾಂಬಿನೇಷನ್ ನೋಡೋಣ, ಜಿನೋಟೈಪ್ ಹೇಗಿದೆ. ಸೊ ಎಕ್ಸ್ಕ್ಲೂಡಿಂಗ್ ದಿಸ್, ನೀವು ಸರಿಯಾಗಿ ಅಬ್ಸರ್ವ್ ಮಾಡಿದ್ರಿ ಅಂತಂದ್ರೆ ದಿಸ್ ಆ್ಯಂಡ್ ದಿಸ್ ವ್ಯಾಲೆಂಟ್, ಇವು 4 ಎಂಟಿಲ್ಲೇ ಅದು ಅಂದ್ರೆ ಈ ರೀತಿ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮಾಡ್ಕೋಬೇಕು. ಎಲ್ ಶೇಪ್ ಬಂದ್ರಿ ಅಂತಂದ್ರೆ ಅದು ವ್ಯಾಲೆಂಟ್ ಆಗಿರ್ತದೆ, ಅಂದ್ರೆ ಡಾಮಿನೆಂಟ್ ಅಲ್ಲಿ ಸಪ್ಲಿಮೆಂಟರಿ ಅಲ್ಲಿ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಆಗಿರ್ತದೆ. ದೆನ್ ಸೆಕೆಂಡ್ ಟೈಪ್ ನೋಡಿ, ಹಿಯರ್ ಈಸ್ 1 capital R. This represents what? Rose comb, but small p recessive, so that rose comb is how many? 1 rose comb. ಹಂಗೆ ಈ ರೋದಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ no rose comb. Here is 1 and here is 1. Totally there are 2. This is 1 rose comb and this is another rose comb. There are 2, and here is 1. This is another one. This is 1 rose comb. Ivu rose comb ಎಷ್ಟು ಆದ ನೋಡ್ರಿ, 3 ಆದವು. ಸೊ ಸೆಕೆಂಡ್. ನೆಕ್ಸ್ಟ್ ಇನ್ನೊಂದು ಸೆಟ್ ಇದೆ. ವಾಟ್ ಈಸ್ ಇಟ್ ನೋಡ್ರಿ, ಕ್ಯಾಪಿಟಲ್ ಪಿ ಇರಬೇಕು, ಸ್ಮಾಲ್ ಆರ್. ಹಿಯರ್ ಈಸ್ 1 and here is 1. ಅಂದರೆ ಕ್ಯಾಪಿಟಲ್ ಪಿ ಬಂತು, ಸ್ಮಾಲ್ ಆರ್ ರಿಸಸ್ ಇದೆ, ಅದು ಕ್ಯಾಪಿಟಲ್ ಪಿ ಇಸ್ ಫಾರ್ ಪಿ ಕೋಂಬ್. ಸೊ ದಟ್ ಇಲ್ಲಿ 2 ಪಿ ಕೋಂಬ್ ಬಂತು, ಪಿ ಕೋಂಬ್. ಓಕೆ, ಆ್ಯಂಡ್ ಈ ರೋದಲ್ಲಿ ಏನಾದರೂ ಬಂತ, ಎಸ್, ಹಿಯರ್ ಈಸ್ 1. ಇಲ್ಲೇನು 1 ಪಿ ಕೋಂಬ್ ಬಂತು. ಉಳಿದದ್ದು ಯಾವ್ದು ನೋಡಿ, ಇದು ಒಂದೇ, ದಿಸ್ ಇಸ್ ಓನ್ಲಿ 1. ಇದು ಒಂದೇ ಒಂದು ಬಂತು, ಇದು ಏನಿದು ಸಿಂಗಲ್ ಕೋಂಬ್. ಸೊ ದ್ಯಾಟ್ ratio by crossing this supplementary genes, 9 we get a ratio 9:3:3:1, where 9 of them are valent, 3 of them are p comb. This is valent, this is p combed fowl, and remaining 3% rose comb and 1% that is a single comb. But what is the difference? Ratio is not modified here, but dihybrid cross carried out by Mendel in case of pea plant, ಅಲ್ಲಿ ಎರಡು ಬೇರೆ ಕ್ಯಾರೆಕ್ಟರ್, ಅದಕ್ಕೆ 2 ಜೀನು. They did not interact, but because they are belonging to the similar locus, belonging to the same locus, ಸೊ ಅಲ್ಲಿ ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ ಎರಡು ಜೀನ್, ಆದರೆ ಇಲ್ಲಿ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಎರಡು ಜೀನ್, ಎರಡು ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಕೂಡಿ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡ್ತಾವೆ ಬಿಕಾಸ್ ದೇ ಆರ್ ಸಪ್ಲಿಮೆಂಟರಿ ಟು ಈಚ್ ಅದರ್. ಆ್ಯಂಡ್ ಬೇಸ್ಡ್ ಆನ್ ದಿಸ್ ವಿ ಕ್ಯಾನ್ deduce or make a statement. Lastly I want to write somewhere here, otherwise you can take down. I will make a statement. What are supplementary factors? 2 marks question ಕೇಳಬಹುದು ಇದನ್ನ. What are supplementary factors? Explain ಅಂದ್ರೆ ಇದೆಲ್ಲ ಬರಿಬೇಕು. This is for totally 10 marks. ಇದನ್ನ ಕೇಳಿದ್ರೆ 2 ಮಾರ್ಕ್ಸ್ ಕೇಳ್ತಾರೆ ಸ್ಟೇಟ್ಮೆಂಟ್ ವಾಟ್ ಆರ್ ಸಪ್ಲಿಮೆಂಟರಿ ಫ್ಯಾಕ್ಟರ್ಸ್ ಅಂತ ಹೇಳಿ. ಅದರ್ವೈಸ್ 10 ಮಾರ್ಕ್ಸ್ಗೆ ವಾಟ್ ಆರ್ ಸಪ್ಲಿಮೆಂಟರಿ ಫ್ಯಾಕ್ಟರ್ಸ್ ಎಕ್ಸ್ಪ್ಲೈನ್ ವಿತ್ ಅನ್ ಎಕ್ಸಾಂಪಲ್ ಅಂದಾಗ ಇದೆಲ್ಲ ಕಂಪ್ಲೀಟ್ ಬರಲಿಕ್ಕೆ ಬೇಕು. ಓಕೆ, ಆ್ಯಂಡ್ ವಾಟ್ ಆರ್ ಸಪ್ಲಿಮೆಂಟರಿ ಫ್ಯಾಕ್ಟರ್ಸ್? Supplementary factors or supplementary genes are the 2 dominant genes where one dominant gene expresses first dominant gene express its phenotype specific its respective phenotype, whereas another dominant gene by interacting with the previous will modify its expression. That is supplementary factor. Again I repeat, supplementary factors or the supplementary genes are the 2 dominant genes where one dominant gene expresses its respective phenotype and the second dominant gene by interacting with the previous will modify its expression. Here in this case, example, see rose comb, capital R for rose comb is dominant, capital P for pea comb is dominant. When capital P reacts with when capital P interacts with capital R, it produced walnut comb. This is the difference. ಎಲ್ಲ ನೋಡಿದ್ರೆ ವ್ಯಾಲ್ನಟ್ ಕೊಂಬ ಇರಲಿ, ರೋಸ್ ಕೊಂಬ ಇರಲಿ, ಪೀ ಕೊಂಬ ಇರಲಿ, ಸಿಂಗಲ್ ಕೊಂಬ ಇರಲಿ, ಕೋಂಬ್ ಪ್ಯಾಟರ್ನ್ ಅನ್ನುವಂಥದ್ದು ಒಂದೇ ಫಿನೋಟಿಪಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್. ಆದರೆ ಎರಡು ಡಾಮಿನೆಂಟ್ ಜೀನ್ ಇನ್ವಾಲ್ವ್ ಅದು, ಯಾಕಂದ್ರೆ ದೇ ಇಂಟ್ರಾಕ್ಟ್ ವಿತ್ ಈಚ್ ಅದರ್, ಬಿಲಾಂಗ್ ಟು ದ ಡಿಫರೆಂಟ್ ಲೋಸೈಡ್. ಆದರೆ ಮೆಂಡಲ್ಸ್ ದ ಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ನೋಡಿದರೆ ಅಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ವೈ ಅನ್ನುವಂಥದ್ದು ಒಂದು, ದಟ್ ಈಸ್ ಫಾರ್ ಕಲರ್ ಆಫ್ ದ ಸೀಡ್, ದೆನ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಅನ್ನುವಂಥದ್ದು ಒಂದು ಶೇಪ್ ಆಫ್ ದ ಸೀಡ್, ಅದಕ್ಕೆ ಕಲರ್ ಆಫ್ ದ ಸೀಡ್ ಅನ್ನು ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗತ್ತೆ, ಶೇಪ್ ಆಫ್ ದ ಸೀಡ್ ಅನ್ನು ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗತ್ತೆ. ಅಲ್ಲಿ ಎರಡು ಫಿನೋಟೈಪ್ ಎರಡು ಜೀನ್, ಇಲ್ಲಿ ಒಂದು ಫಿನೋಟೈಪ್ ಎರಡು ಡಾಮಿನೆಂಟ್ ಜೀನ್ಸ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಅಂಡ್ ಕ್ಯಾಪಿಟಲ್ ಪಿ. ಸೊ with this we can prove as well as understand what do you mean by supplementary factors and what is their classical ratio, that is 9:3:3:1 belonging to the same phenotype as a comb pattern. Okay, and secondly that is ratio is like this, and the meaning or the statement for supplementary factors already I told you, and example, that is entire this thing you have to explain both. So question mark ಇಟ್ಟಿದ್ದೆ, ಆ question mark ಗೆ ನೀವು ಏನು ತುಂಬಬೇಕಾಗತ್ತೆ ಅಂತಂದ್ರೆ walnut comb. When do you get walnut comb? When you cross pea combed fowl with rose combed fowl. Okay, I hope you people followed what all I explained so far as a first concept in, that is interallelic or intergenic interaction. And in my next session I am going to open one more new concept, that is the complementary factors, in which we get the classical ratio, that is 9:3:3:1 will get modified. How is that? We'll see it in the next session. I hope you are fine and fit at home, and please stay away from the COVID infection and COVID infection free, and please do work by reviewing this video which I am going to upload very shortly today or tomorrow. Please view it, and if you get difficulties or questions, please put me questions in WhatsApp group. Thank you.